Hello students, welcome to George Biology. In the previous sessions, we have gone through many different structures in the eukaryotic cell. In today's session, we'll go with cytoskeleton. If you look at a cell, in that cell, due to presence of many things, the shape of the cell that can be maintained. It might be cell wall, it might be cytoskeleton, right? So here, okay, let's say if you talk about plant cell. In the plant cell, very comfortably looking at the structure itself, you can say it has a cell wall due to which it is able to maintain its shape. But if the cell wall is not there, even though the cell that can maintain the shape, and that is because of cytoskeleton. Now what is cytoskeleton? Cytoskeleton, it's a network of elaborate filamentous proteinaceous structures that helps in maintaining shape of the cell. So what is a cytoskeleton? It's a minute, fibrous, proteinaceous structures that helps in maintaining shape of the cell. And it also helps in providing mechanical support. Mechanical support. Along with this one, it helps in motility of the cell. It consists of three different components. Altogether, we call them as cytoskeleton. Among them, the major ones, that is microtubules. From the point of NCERT, it's a small topic, but here in the exam, a lot of different connections that can be given, due to which we are going to elaborate this topic a little bit. So here, microtubules. Microtubules, they are the major cytoskeleton members. And these microtubules, they are made of a specific protein that is called as tubulin protein. It's a tube-like structure. In the name itself, you can find it. It's a tube-like structure. And this one, if you cut it and if you observe its transverse section, it will be looking like this, where it consists of 13 protofilaments. All together they join and helps in formation of a microtubule. It means a complete microtubule that consists of how many protofilaments? 13 filaments that are present. And if you look at the inner diameter, that is about 15 nanometer, and the outside, it is 25 nanometer of diameter that is present. Now these protofilaments, they are helping in formation of microtubule, we said. If they want to form a complete tube by joining, there should be some material which can join them all together. Now this association, or is the assembly, the association is due to presence of GTP, guanosine triphosphate. Number 2, an element is required, that is calcium. And number 3, calmodulin is required. Means when you go with mineral nutrition, you can get a question. The element which is required for the formation of microtubules, that is calcium, is required. Now this microtubule, what we said, it is made of 13 protofilaments. 13 protofilaments. How they are jointed? They are joined parallelly. They are joined parallelly, side by side, for making what? For making a hollow tube. And this hollow tube, whichever is made, that is called as microtubule. And this microtubule, it is made of a specific protein which is non-contractile. Non-contractile protein that is involved. So what we said, it is made of 13 protofilaments, we said. So here if you look at the 13 protofilaments, and these 13 protofilaments, they are made of a protein which is called as alpha and beta tubulin protein. Alpha and beta tubulin protein is utilized. Let's look at the function here. Now if you look at the function, see here, as per the NCERT, if you're observing, the required functions we have written already on the top. Here you can see over the top, 3 functions are there in the NCERT. Exactly, we placed it over the top. But with respect to the microtubule, if any further applications are given, for that reason, we are taking this one additionally. So if you look at the functions, this helps in formation of mitotic filaments. It means if microtubules are there, mitotic filaments will be formed. If mitotic filaments are formed and if the mitosis is taking place by formation of microtubules, then only we call it as mitotic binary fission. If it is not there, amitotic binary fission. As simple as that. Next one, spindle apparatus. It helps in formation of spindle apparatus. It helps in formation of cilia and flagella. So here itself we are getting a clear point. The protein which is involved in formation of cilia and flagella in eukaryotes, that is tubulin protein. It means if tubulin is there, microtubule will be there. If microtubule formation occur, cilia and flagella formation takes place in eukaryote. That makes the difference between prokaryotic flagella and eukaryotic flagella with respect to the composition. Next, it helps in formation of astral rays. Astral rays. When we go with cell cycle, you will come to know what is this astral ray. Next one. Because of these mitotic filaments, during the time of cell cycle, they help in pulling the chromosomes towards the side of poles. It means they provide anaphasic movement of chromosomes during the time of cell cycle. And the last one, it helps in formation of centrioles. Now this is about microtubule, and I already said why microtubule we have to elaborate it more. Why? Because at many places its application will be asked. And this is the major cytoskeleton member. Number B, the second cytoskeleton member, that is microfilament. Microfilaments. If you look at the microfilament, it is solid, rod-like structure, and it is contractile in nature. It is made of contractile protein, and the type of protein which is present over here, that is two different type of proteins are present. Number one, actin, and number two, myosin. And this actin, it's a globular protein, and here myosin, it is a filamentous protein. Filamentous protein. Let's look at the functions of this microfilament. If you look at the function, as it's a contractile fiber, it can contract and bulge towards outside, or else it can contract and invaginate towards inside. If it bulge towards out, it helps in formation of pseudopodia. Pseudopodia formation is due to the activity of microfilament. If it invaginates inside, that helps in formation of cell furrow, due to which it promotes cell furrow method of cytokinesis. So it helps in cell furrow formation, and that is helping in cytokinesis. Now it consists of ATP synthase activity. It consists of ATP synthase activity. If a question is asked, the type of cytoskeleton member that consists of ATP synthase activity, that is microfilaments. That is how you can take the question. We'll go for the last and the minute aspect of cytoskeleton, that is intermediate filament. It is made of acidic protein. Usually you will not get any question from this point of discussion of intermediate filament. That's why we are keeping it very simple. It is non-contractile in nature, and it helps in formation of scaffold or sac-like structure around the nucleus. Scaffold or else sac around nucleus. Let's take some questions from the topic which we have discussed. Which is a major cytoskeleton member? That is microtubule. What is the composition of cilia and flagella in eukaryotes which makes this one different from prokaryotic flagella? That is tubulin protein. Out of the different cytoskeleton members, which is contractile in nature, or else which is made of contractile protein? What is the answer? Microfilaments is the suitable answer. Out of the following, which is odd with respect to the functions of microtubules? Number one, It helps in formation of mitotic filaments. Number 2, it helps in formation of cilia and flagella in prokaryotes. Number 3, it helps in formation of astral rays. And number 4, it helps in formation of spindle apparatus. What is the suitable answer here? Prokaryotes is the one where flagella is made of flagellin protein, not tubulin protein. Now that's a suitable answer. In the next session, we'll go with centrioles.